Understanding the scientific method and experimental design. Welcome and overview. Welcome to ATI TEAS 7 Science Series, where we explore scientific reasoning. In this video, you will learn how to use logic and evidence to evaluate and solve problems, the steps of the scientific method, how to design and conduct experiments, how to collect, analyze, and interpret data, the difference between theories and laws, ethical considerations in scientific research. Introduction to scientific reasoning. Scientific reasoning is a systematic approach to understanding the world through observation and evidence. What is scientific reasoning? The ability to use logic and evidence to evaluate claims and solve problems. A critical thinking skill essential for healthcare professionals. The foundation of evidence-based medicine. Why is it important? Helps healthcare professionals make informed decisions. Enables you to evaluate medical research and studies. Allows you to distinguish between valid and invalid claims. Supports problem-solving in clinical settings. Applications in healthcare. Interpreting lab results. Evaluating treatment effectiveness. Understanding research studies. Making evidence-based clinical decisions. The scientific method. The scientific method is a systematic approach to investigating problems and finding solutions. Definition: a standardized process used by scientists to investigate phenomena, acquire new knowledge, and correct previous understanding. The steps of the scientific method. 1. Observation. Identify a problem or phenomenon to study. Notice patterns or unusual occurrences. Example: patients with certain symptoms respond differently to treatment. 2. Question. Formulate a clear, specific question based on observations. Example: does body temperature affect medication absorption? 3. Hypothesis. Develop a testable prediction. An educated guess about the answer. Must be specific and measurable. 4. Experiment. Conduct tests to gather data. Follow a controlled procedure. Collect observations and measurements. 5. Analysis. Interpret the results. Look for patterns in the data. Use statistics and graphs. 6. Conclusion. Accept or reject the hypothesis based on evidence. Determine if results support or contradict predictions. Share findings with others. Identifying a research question. A well-formulated research question is essential for good science. Characteristics of a good scientific question. Specific and focused. Narrow enough to be answerable. Clear about what is being investigated. Measurable. Can be answered through observation and data collection. Variables can be quantified. Based on observations. Stems from real-world phenomena. Addresses gaps in current knowledge. Answerable through experimentation. Can be tested using scientific methods. Not based on opinion or values. Examples. Poor questions. Why is the sky blue? Too broad, already answered. Is chocolate the best food? Based on opinion. What is the meaning of life? Not scientifically testable. Good questions. How does temperature affect enzyme activity in saliva? Does caffeine consumption affect heart rate in adults? What is the relationship between sleep duration and test performance? Formulating a hypothesis. A hypothesis is a testable statement that predicts the outcome of an experiment. Definition: a proposed explanation for a phenomenon that can be tested through experimentation. Characteristics of a good hypothesis. Makes a specific prediction. Is testable and falsifiable. Based on prior knowledge or observations. clearly identifies the relationship between variables. Hypothesis format. If, then, because. Structure. If introduces the independent variable, what you change. Then predicts the dependent variable, what you measure. Because provides reasoning based on scientific knowledge. Examples. If plants receive more sunlight, then they will grow taller, because photosynthesis increases with light exposure. If students study for longer periods, then their test scores will increase, because repetition improves memory retention. If water temperature increases, then sugar will dissolve faster, because molecular movement increases with heat. Experimental design variables. Understanding variables is crucial for designing valid experiments. Types of variables. Independent variable. The factor that the experimenter deliberately changes or manipulates. The cause in a cause-and-effect relationship. Only one independent variable should be tested at a time. Example, amount of fertilizer given to plants. Dependent variable. The factor that is measured or observed. The effect in a cause-and-effect relationship. Changes in response to the independent variable. Example, plant height or growth rate. Control variables, constants. Factors that are kept the same throughout the experiment. Ensure a fair test by eliminating other influences. Allow you to attribute changes to the independent variable alone. Example, type of soil, amount of water, temperature, light exposure. Example experiment, testing how light affects plant growth. Independent variable, amount of light exposure. Dependent variable, plant height. Control variables, soil type, water amount, temperature, plant species, pot size. Experimental and control groups. Well-designed experiments require both experimental and control groups for comparison. Experimental group. The group that receives the treatment being tested. Exposed to the independent variable. Provides data about the effect of the treatment. Example, patients receiving a new medication. Control group. The group that does not receive the treatment. Used as a baseline for comparison. Shows what happens without the experimental treatment. May receive a placebo, inactive substance. Example, patients receiving a sugar pill instead of medication. Why control groups are important. Eliminate alternative explanations for results. Account for placebo effect. Provide a standard for comparison. Strengthen the validity of conclusions. Example in medical research, testing a new blood pressure medication. Experimental group, 100 patients receive the new drug. Control group, 100 similar patients receive a placebo. Compare blood pressure changes between both groups. Collecting and analyzing data. Accurate data collection is essential for drawing valid conclusions. Types of data. Qualitative data. Descriptive observations. Uses words, not numbers. Describes qualities or characteristics. Examples. Color of a solution. Texture of a substance. Smell or taste. Patient's symptoms descriptions. Quantitative data. Numerical measurements. Can be counted or measured. Allows for statistical analysis. Examples. Length, height, weight. Temperature. Time. Heart rate, blood pressure. Importance of accurate data collection. Reduces errors, minimizes mistakes that could invalidate results. Ensures reliability, allows others to replicate the experiment. Supports valid conclusions, provides strong evidence for or against the hypothesis. Enables statistical analysis, quantitative data can be analyzed mathematically. Best practices. Use calibrated, accurate instruments. Take multiple measurements. Record data immediately and clearly. Include units of measurement. Note any visual representation of data makes patterns and trends easier to identify. Common types of graphs. Bar graph, bar chart. Used to compare different groups or categories. Shows discrete data. Bars can be vertical or horizontal. Example, comparing test scores of different study groups. Line graph. Used to show trends over time. Displays continuous data shows how variables change. Example, temperature changes throughout the day, plant growth over weeks. Pie chart. Shows parts of a whole. Displays percentages or proportions. All sections add up to 100%. Example, distribution of blood types in a population. Scatter plot. Shows relationship between two variables. Each point represents one data pair. Reveals correlations or patterns. Example, relationship between study time and test scores. Choosing the right graph. Consider the type of data you have. Think about what you want to communicate. Match the graph type to your purpose. Ensure the graph is clear and easy to read. Drawing conclusions. After analyzing data, scientists draw conclusions based on evidence. Steps in drawing conclusions. 1. Compare results to hypothesis. Do the findings support or reject your hypothesis? Look at the data objectively. Consider both expected and unexpected results. 2. Analyze patterns. Identify trends in the data. Look for relationships between variables. Consider the strength of the evidence. 3. Consider alternative explanations. Could other factors explain the results? Were there any errors or limitations? What confounding variables might exist? 4. State your conclusion clearly. Summarize what the data shows. Refer back to the original question. Be specific about what was learned. Possible outcomes. Hypothesis supported, data confirms the prediction. Hypothesis rejected, data contradicts the prediction, this is still valuable. Inconclusive results, data is unclear or inconsistent. Next steps. Modify the experiment if needed. Conduct further testing. Share findings with the scientific community. Develop new questions based on results. Scientific theories versus laws. Theories and laws are both important in science but serve different purposes. Scientific theory explains why something happens. A well-substantiated explanation based on extensive evidence. Can change with new evidence. Supported by many observations and experiments. Examples. Theory of evolution. Cell theory. Germ theory of disease. Atomic theory. Important note, in science, theory doesn't mean guess, it represents our best understanding based on evidence. Scientific law. Describes what happens. States a consistent pattern observed in nature. Often expressed as mathematical formulas. Does not explain why the pattern occurs. Examples. Newton's laws of motion. Law of conservation of energy. Law of gravity. Key differences. Theories explain mechanisms and processes. Laws describe observable patterns. Both are based on evidence and testing. Laws are more universal and unchanging. Theories provide deeper understanding. Measurement in science. Accurate measurement is fundamental to scientific investigation. The SI system, International System of Units. Standard units used worldwide in science to ensure consistency. Common SI units. Length meter, m. Centimeter, cm, millimeter, mm, kilometer, km. Mass kilogram, kg. gram, g, milligram, mg. Volume liter, L. Milliliter, ml, cubic centimeter, cm superscript 3. Temperature Kelvin, K, or Celsius, degrees C. Fahrenheit, degree F, used in some countries but not standard in science. Time second, S. Minute, min, hour, H. Why the SI system is important. Ensures consistency across all scientific research. Allows scientists worldwide to communicate clearly. Facilitates comparison of results. Reduces confusion from different measurement systems. Essential for replicating experiments. Conversions. Scientists must be able to convert between units. Example, 1 kilogram equals 1,000 grams. Example, 1 liter equals 1,000 milliliters. Accuracy versus precision. Understanding the difference between accuracy and precision improves data quality. Accuracy. How close a measurement is to the true or accepted value. Relates to correctness. Can have one accurate measurement. Example, if the actual temperature is 25 degrees Celsius, a reading of 25.1 degrees Celsius is accurate. Precision. How consistently measurements can be repeated. Relates to reproducibility. Multiple measurements clustered together. Example, taking 5 measurements that all read 23.5 degrees Celsius shows precision. The 4 possible scenarios. 1. High accuracy, high precision. Measurements are close to the true value and consistent. The ideal situation in science. 2. High accuracy, low precision. Measurements average near the true value but vary widely. Results are scattered but centered correctly. 3. Low accuracy, high precision. Measurements are consistent but wrong. Systematic error present. Example, a scale that always reads 2 kilograms too high. 4. Low accuracy, low precision. Measurements are both inconsistent and incorrect. The worst scenario indicates major problems. Target analogy. Accuracy equals hitting the bullseye. Precision equals all arrows grouped together. Best result equals all arrows grouped in the bullseye. Common errors in scientific experiments. All experiments have potential sources of error that can affect results. Types of errors. Human error. Mistakes in reading instruments. Errors in recording data. Miscalculations. Carelessness or fatigue. Example, misreading a thermometer or recording the wrong number. Instrument error. Faulty or uncalibrated equipment. Equipment limitations. Wear and tear on instruments. Example, a scale that isn't zeroed properly, a broken thermometer. Sampling error. Using too small a sample size. Biased sample selection. Non-representative samples. Example, testing a drug on only 10 people instead of 1,000. Environmental error. Uncontrolled variables affecting results. Changes in temperature, humidity, or other conditions. External factors not accounted for. Ways to reduce errors. Use properly calibrated equipment. Take multiple measurements and average them. Increase sample size. Control environmental conditions. Follow standardized procedures. Have multiple researchers verify data. Use appropriate precision for measurements. Repeat experiments for reliability. The role of science in healthcare. Scientific reasoning and research are fundamental to modern healthcare. Examples of scientific reasoning in medicine. Drug testing and development. Clinical trials follow the scientific method. Phase 1, 2, and 3 trials test safety and effectiveness. Double-blind studies prevent bias. Data analysis determines if drugs should be approved. Medical diagnostics. Doctors use symptoms, observations, to form hypotheses. Order tests, experiments, to gather data. Analyze results to reach a diagnosis, conclusion. Adjust treatment based on patient response. Evidence-based practice. Medical decisions based on scientific research. Uses the best available evidence. Combines clinical expertise with patient values. Continuously updated as new research emerges. Public health research. Epidemiological studies track disease patterns. Identify risk factors and protective factors. Guide policy decisions and interventions. Examples: vaccine development, disease prevention strategies. Quality improvement. Hospitals use scientific methods to improve care. Collect data on patient outcomes. Test new protocols and procedures. Measure effectiveness and make adjustments. Ethical considerations in science. Ethical guidelines protect research participants and ensure integrity in science. Key ethical principles. Informed consent. Participants must understand the research before agreeing. Must be told about risks and benefits. Can withdraw at any time without penalty. Must be voluntary, not coerced. Special protections for vulnerable populations. Bias and objectivity. Scientists must avoid letting personal beliefs influence results. Use blind and double-blind studies when possible. Report all findings, even those that contradict hypotheses. Disclose conflicts of interest. Peer review helps maintain objectivity. Animal and human research ethics. Minimize harm to participants. Benefits must outweigh risks. Use the minimum number of subjects necessary. Institutional review boards, IRBs, approve studies. Follow established guidelines, Helsinki Declaration, Belmont Report. Research integrity. Never falsify or fabricate data. Give credit to others' work, avoid plagiarism. Report methods accurately so others can replicate. Correct errors when discovered. Share data and results with the scientific community. Special considerations. Extra protections for children, prisoners, and vulnerable groups. Cultural sensitivity in international research. Privacy and confidentiality of participants. Fair selection of research subjects. Summary and key concepts. Let's review the essential principles of scientific reasoning we've covered. Key concepts. The scientific method provides a systematic approach to investigation. Well-designed experiments require clear hypotheses and controlled variables. Independent variables are manipulated, dependent variables are measured. Control groups provide baseline comparisons for experimental groups. Data can be qualitative, descriptive, or quantitative, numerical. Graphs and charts help visualize patterns and trends in data. Conclusions must be based on evidence, not assumptions. Theories explain why, laws describe what happens in nature. Accuracy measures correctness, precision measures consistency. Understanding sources of error helps improve experimental design. Scientific reasoning is essential in healthcare for evidence-based practice. Ethical guidelines protect research participants and ensure integrity. Remember, scientific reasoning isn't just for laboratories, it's a critical thinking skill you'll use throughout your healthcare career to evaluate information, solve problems, and provide the best possible patient care. Congratulations! You now have a comprehensive understanding of how to apply the scientific method, design experiments, analyze data, and think critically about evidence. You've completed all 4 modules. Module A, Human Anatomy and Physiology. Module B, Biology. Module C, Chemistry. Module D, Scientific Reasoning. Next steps. Review all 4 modules to reinforce your learning. Practice applying these concepts to TEAS-style questions. Use scientific reasoning to evaluate healthcare information. Connect concepts across all modules to see how they relate. Final thoughts. You've built a strong foundation in the sciences essential for healthcare. The knowledge you've gained will serve you well on the ATI TEAS 7 exam and throughout your career in healthcare. Your dedication to learning demonstrates the scientific curiosity and critical thinking skills that make excellent healthcare professionals. Keep studying, stay curious, and trust in the preparation you've done. You've got this. Best of luck on your ATI TEAS 7 exam and in your healthcare career.